Welcome once again to this online lecture on neuroanatomy series. In this particular lecture, we are going to look at the limbic system. Maybe you've heard about limbic system many times when you're looking at brain. I just thought it'd be nice to have a consolidated lecture on this particular system. We begin by just a matter of introductory concepts, perhaps by just defining the origin of the term or trying to understand more about the origin of the terminology. So the person who coined the term limbic system derived this term from its key meaning, which means bordering or edge. The structures that he noted to be forming part of this system appeared to be edging or bordering the cerebral hilum, particularly the cortical tissue around this particular region. So in the shaded region, you see some cortical material that seems to form a rim around the cerebral hilum. This is what he called the limbic system, or the limbic lobe, so to speak. When you talk of cerebral hilum, we're referring to these structures that join the right and the left hemisphere. So basically, the region around the corpus callosum and the region around the dencephalon. So the limbic cortex, as it is, were the structures that are around the cortical tissue that are around the corpus callosum and the dencephalon. Functionally, the limbic system is not limited to that cortical tissue only. It also extends to cover the subcortical nuclei, which connect these particular regions. So there are a number of subcortical nuclei with some specific functions that are also part of the limbic system. In this particular image, you can perhaps see a few like that one pointed there, which is the mammillary body of the hypothalamus. The hypothalamus itself is actually a nucleus on its own or a group of nuclei. The thalamus, a group of nuclei. So there are many nuclei with limbic functions. In this particular image, you can see the thalamic nuclei. Yes, you can see the mammillary bodies. Perhaps you can see the basal nuclei and the amygdala nuclei there. They are still part of the subcortical nuclei. In this particular image, there are more nuclei you can see. For example, the septal nuclei there, among others that we've already mentioned. Now, again, it's not limited to the nuclei and the cortical tissue only. It is also involving some white matter tracts that join the nuclei and the cortices. So we have several axonal fibers that join the nuclei and the cortices. They're also part of the limbic system. So therefore, currently, when we talk of the limbic system, we are not restricting ourselves to these structures which are in the hilum or around the hilum of the cerebrum, but we extend it to just include the entire neuronal circuitry that tend to control emotions and behavior and also involved in motivational drives. So that's the current concept. In this particular lecture, therefore, we are going to learn the following. First, we look at the structural and functional characteristics of the components of the limbic system. There's some structural characteristics which are important to note that I want us to highlight. We'll then take time to explain the key functions of the limbic system. You may have heard of many others, so we'll just consolidate them here. We'll take more time to just look at the components of the limbic system. The different components, we'll outline them one by one and talk about them. And then we'll explain some major functional circuits which are in the limbic system that help to perform its functions. And lastly, we'll highlight some clinical conditions which are related to the limbic system, that disorders which are related to the limbic system. So let's begin with the characteristics of the components of the limbic system. The limbic structures have some key characteristics, and maybe some three to note here. One is that the limbic system contains what we call allocortices. Allocortices refer to cortical tissue that has less than 6 layers. Perhaps you already know that there's something called the neocortex. The neocortex, the cortical tissue that has 6 layers histologically. And in another setup, you may have named those 6 layers. Now we're saying there's some regions of the brain that do not necessarily have those 6 layers, so they're not considered neocortical. Therefore, we call them allocortical. There's 2 varieties of allocortex. There's the archicortex, which is a 3-layered cortex and viewed as a bit primitive, and a paleocortex, which is a transitional cortex made up of about 4 to 5 layers histologically. So structures of the limbic system, key structure of the limbic system have these allocortical layers. Second important thing to note is that we have evidence of prolonged after discharge within the circuits of the limbic system. This is to mean that when some neurons are stimulated in one or another, the effect of that stimulus takes quite a long time revolving within the neurons of the limbic system. And that seems to suggest, therefore, that perhaps this is one of the mechanisms that the limbic system uses in terms of retaining memory. Perhaps also explains why emotional experiences take long to be abolished, because of this evidence of prolonged after discharge. Last but not least, is that limbic system, although it has some input from the neocortex, there is little input from the neocortex. That seems to suggest that it's possible that limbic system is relatively independent, that it may function without the neocortex. Having said that, we also know a very fundamental truth about the limbic system, that although this neocortical input is little, it's actually very important in the function of the limbic system, because it can actually override the function of the limbic system. That neocortical input can influence the limbic system. And again, that is important to note. We know that neocortical function can actually influence some emotional drive that we have. Therefore, these are the key characteristics of the limbic system I wanted us to highlight. With that in mind, let's now outline the key functions of the limbic system. Well, I've chosen to pick some key functions and then others. Let's, on this particular slide, highlight those key functions. So first, we know that limbic system is very important in learning and memory. Key structures within the limbic system are important in processing new information that comes in. And they can choose to reinforce this new information, which means help to develop memory for it. Or they may choose to ignore it. So it doesn't matter when if it comes several times, there's no much response, and what we call habituation. And you realize that may not therefore trigger much of memory storage. And so limbic system is important in processing information that are important for memory, for reinforcement. Very key. 2, tailored to that is the emotional control, and perhaps this is how we know the limbic system. It tells us to create emotional responses such as fear, rage, or placidity. These are responses that we are developing from various inputs that have come to us. maybe in our interaction with people and with the environment, we develop these emotional responses. Now, the response that limbic system creates may also be linked with some visceral motor responses. This visceral motor response can be mediated by the autonomic nervous system, especially sympathetic nervous system or parasympathetic system. But we also have evidence of limbic system influencing endocrine output, influencing hypothalamic function that control hormones. And so there's some responses that we develop which have an endocrine and the autonomic components. For example, you may have an experience with something and you respond in fear and even your blood pressure goes up, for example, or you start sweating because of that experience. You know, that's an autonomic response, but it's tailored with the emotions that we have. Third, limbic system is known to be involved in motivation and addiction behaviors. This is because it has some reward circuitry within it that cause excitation. So these circuits, when they're stimulated, there'll be some excitations. And because of that, you might want to look forward to another event. And so it helps you even to develop some cues. When you see something, you get a relapse. And because of this, it has been associated with addictive behaviors. Sexual behaviors are also controlled in the limbic system as part of emotional responses, but also we know that limbic system receives some sensory input from genitalia, and it provides responses which are consistent with sexual excitement and perhaps libido and things like that. Limbic system also controls our nutrition, food intake, to be specific, because it has a capacity to control appetite and even eating behavior, like the choice of food you want to take, whether you feel like you're now eaten enough and things like that. Limbic components have these functions. I consider these 5 key functions, but the list is not over. There are still other functions. For example, social cognition. Limbic system is helpful in helping us develop thought processes which are involved in understanding and also dealing with people, how you interact with people socially, how you integrate or interpret those experiences and what it makes you feel and the choices you make about them. There have also been evidence to suggest that limbic system interweaves our unconscious emotions with our cognitive thoughts and perceptions. And perhaps through this, we're able to link emotions and memory during sleep. And perhaps this also contributes to the dream experiences that we do have. There's some vegetative functions that could also be influenced by the limbic system. This is perhaps because these inputs that we have an effect on the hypothalamus, and hypothalamus has effect in all these areas. So perhaps because of that, we can say that limbic system can influence some vegetative functions like temperature control, osmolarity, appetite. Osmolarity will be controlled largely by the endocrine responses. Olfaction is an important function in the body. Structures which have olfactory functions seem to be connected with the limbic system. And initially, this was actually even thought that a big system was actually called rhinencephalon, because it was thought that all these things have olfactory functions because of the connections. With further knowledge, we've noted that that's not exactly true. There might be actually just separate things. However, key structures with limbic functions still receive input from olfaction, and important olfactory processes occur within these structures. And so we can still link olfactory functions to limbic functions. There could be a link between how is the emotional responses to smell and creating olfactory memory. They're still important in that sense. So we've outlined the key functions of the limbic system. I want us to now take more time to look at the components. Sorry, we've outlined the key functions of the limbic system. Now I want us to take more time to look at the components of the limbic system. I choose to divide the components of the limbic system into three. That we have the nuclei associated with the limbic system, we have the limbic lobe, and we have the fiber tracts. If we're to say something about the nuclei, and we'll take time to look at each of them, they're the subcortical aggregation of neuronal cell bodies. They are nuclei, which are associated with the limbic lobe. And remember, the fiber tracts are just nerve pathways. They are neuronal pathways that connect various nuclei and cortices of the limbic system. Now, coming back to this central region, the limbic lobe, we can divide the limbic lobe into two. We have what we call the limbic cortex. The limbic cortex is the actual rim of cortical tissue that's around the cerebral hilum that warranted or that made the first person to coin the term limbic actually call this system limbic because of that cortical material at the rim of the cerebral hilum. So we look at that limbic cortex. As we know it today, maybe that has extended beyond the rim. We'll talk about that. And then we have what we call hippocampal formation. Hippocampal formation are some structures located within the medial aspect. The term mesial means medial. The medial aspect of the temporal lobe. Indeed, the temporal lobe houses a lot of structure of the limbic system, and especially hippocampal formation. And so we'll also have a look at that. First, I want us to begin our talk with the limbic lobe, and in particular, the limbic cortex. So by definition, we already said that rim of cortical tissue around the cerebral hilum, and this shading has made it easier for us to see that rim. You can see it's almost taking the letter C or ovoid kind of. This cortical tissue become, therefore, the cortical association areas for emotions. When you look at functions of the cerebral cortex, you notice that we have association cortices. And so the limbic cortex are actually association areas for emotions. There are quite a number of components. So we have what you call the betofrontal cortex, which is located slightly lower, not shown in this particular image. We have the subcallosal area there. We have the cingulate gyrus, which is this one. We have the parahippocampus, which is that one. Occasionally, you may see something here called the isthmus. It's just the narrow junction between the cingulate gyrus and parahippocampal gyrus, as you can see there. And then we have the insular cortex. So insular cortex is not shown in this particular image. It's actually not forming part of that lead curve or rim, but we know that insular cortex also has limbic functions, so we'll also talk about it. In general, the limbic cortex function as a transition zone through which signals are transmitted from the neocortex to the limbic system. And so let's have a look at each of the components of the limbic cortex, beginning with the orbitofrontal cortex. Orbitofrontal cortex is located in the inferior surface of the frontal lobe, so not shown in this one, but we can see it here. So that's the orbitofrontal cortex. This is a lateral view of the left cerebral hemisphere. So that's the location. If you have an inferior view of the brain, then this is the orbitofrontal cortex. if you are to put this in anatomical position, we'll notice that that part of the frontal lobe actually rests on the roof of the orbit within the anterior cranial fossa. In terms of key functions, especially linked to limbic, we know that the orbitofrontal cortex is involved in the cognitive processes that help in decision making. So it's an important part of the limbic system. It's also important in processing reward behaviors. So just processing some reward behaviors. Second is the subcallosal, or what we call para-olfactory area. And perhaps we take note that we're using the term area and not gyri or gyrus. There's some not so much confluence in terms of understanding the terminologies. There's understanding that the subcallosal gyrus is actually a different thing from subcallosal area. So let's use the term subcallosal area. The person who described it called it the Zuckerland's gyrus. So it's a bit different from the subcallosal gyrus itself. The subcallosal area is located in the medial surface of the frontal lobe, just below the rostrum of the corpus callosum. So if this is the corpus callosum, remember this is the genu of corpus callosum. That's the rostrum of corpus callosum. This is the body, and this is the splenium of corpus callosum. So we're talking about below the rostrum of corpus callosum, which is here, and it's anterior to the paraterminal gyrus. So paraterminal gyrus, that small stripe there. The paraterminal gyrus is actually what now many people consider the subcallosal gyrus. Yet the subcallosal area is a bit expansive and anterior to that region. In terms of functions, there's a link between the subcallosal area and depression. People with depression may have some firing or increased activity in this area. And so we know that the subcallosal area has involvement in development of negative mood. But we also know that this area is also important in regulation of some autonomic responses, perhaps related to these negative moods. We can now talk about the cingulate gyrus, a very big gyrus on top and around the corpus callosum. It actually forms a curve around the corpus callosum. The cingulate gyrus, the cortex within it, receive input from the neocortex. So there are connections from the rest of the neocortex to the cingulate cortex. A lot of connections from this side to here, from that side to there, from that side to there. And this information, the cingulate cortex will process and also send signals to the hippocampal formation. Cingulate cortex has a role in emotion formation and processing, so just forming that emotion and processing that emotion. It also has a role in learning and memory, maybe because of even the fibers it contains within. It also has a role in autonomic functions, and some people will also add that has a role in pain processing. So quite a lot of functions associated the cingulate gyrus. In terms of the disorders, we see that people with depression actually, or let me put it this way, depression and schizophrenia has actually been linked with disorders in the cingulate gyrus. So we're not saying that this is a cause, but there's some linkage, there's some implication. As you're going to see in diseases like schizophrenia, there could be multiple parts of the brain which are involved, not just one part. And so perhaps cingulate is one of those many parts which are involved in the disease process. The other gyrus talk about is the parahippocampal gyrus, located in the inferior middle aspect of the temporal lobe. So this is the parahippocampal gyrus, continuous with the cingulate gyrus through its isthmus there. Parahippocampal gyrus provides a major path by which sensations, or let's say information from the rest of the brain, converge to the hippocampus. It's an important part for that because hippocampus will be somewhere here, so the information come through there. It is involved in encoding and recognizing social and environmental scenes. It's able to encode some environmental scenes. It's also able to encode some social scenes, for example, how people behave around you, how they behave, and what that actually means. All right, we can now talk about the insular cortex, which is not part of the cortical rim around the hilum, but still part of the limbic cortex. So insular cortex lies deep in the floor of the Sylvian fissure. Remember Sylvian fissure, the latus sulcus. It is overlapped by the operculum of the frontal lobe, operculum of the parietal lobe, and operculum of the temporal lobe. So perhaps you need to open to see the insula with some short gyri and long gyri, and a sulcus going around it, what we call the circular sulcus. The insular cortex is important in playing a role in diverse functions, which are actually linked to emotions and homeostasis. In particular, we know that insular cortex is important in processing convergent information that help to produce subjective emotions that are relevant in a particular context. So there are subjective emotions, which means you may interact with somebody or something and you choose to be very angry, but somebody else will not be very angry with that particular kind of exposure. So they're very subjective. Or development of, you just feel happy about something and somebody else may not actually have the same experience in that context. Other functions of the insular cortex could be regarding autonomic functions, regarding taste, because we know that the taste area is very near there. Regarding hearing, again, because the primary auditory area is very near here, actually on the transverse temporal gyri there. Autonomic functions also be linked to the insular cortex, and salience, maybe just something about uniqueness, something that makes something outstanding. Okay, so remember, we are still talking about components of the limbic system, and we are discussing limbic lobe. We have just talked about the limbic cortex, which is the rim of cortical tissue around the cerebral hilum. I put a star there to remind you that the insular cortex is not part of that particular rim, but yes, still part of the limbic cortex. The other component of the limbic lobe is hippocampal formation, which refer to some structures within the mesial temporal lobe. And I want us to now look at the hippocampal formation. So hippocampal formation are structures within the medial aspect of the temporal lobe. Constitute the following. We have hippocampus, dentate gyrus, entorhinal cortex, subiculum. So in this image, that's the hippocampus, and deep to it, we have the dentate gyrus there. This is a view from the top with the parts which are supposed to be covering this thing have been removed. Parietal lobe has been removed. We are looking at the lateral ventricle, the inferior horn of the lateral ventricle in the floor, and that's what we are seeing, the hippocampus and the dentate gyrus there. Whatever we're calling subiculum may have, it's part of a big thing, what we call subicular complex, which contains subiculum itself, presubiculum and parasubiculum. Now, these structures are usually folded and rolled up, hence submerged deep into the parahippocampus. Let's look at this. These are coronal section through the temporal lobe, and this is the right one, and we're looking at it from the front. So this is the parahippocampus. We can see the rolling there, that cortical tissue rolling there like that. So this is actually the formation, that rolled up cortical tissue is the hippocampal formation. In particular, so this region here will represent the entorhinal cortex. It's usually on the hippocampus. Then you have this region there, the subicular complex. Then this the hippocampus. And the last component is the dentate gyrus, which is this one. On this particular image, you can still trace that. So this is part of parahippocampus, and parahippocampus houses the entorhinal cortex. Following that, this is the subicular complex, that region. Then beyond that, this is the hippocampus. And then the last component of that, the dentate gyrus. This image, again, the same, the parahippocampus, so entorhinal cortex will be here, then subicular, then hippocampus, and lastly, the dentate gyrus. Now, tailored to the hippocampal formation are some things we call hippocampal rudiments, which are basically vestigial extratemporal extensions of the components of the hippocampal formation. And you'll also look at them. Let's have a look at each of the components of the hippocampal formation, beginning with the entorhinal cortex. So the entorhinal cortex, as I've already mentioned, is located on the parahippocampus, actually in the anterior part of the parahippocampus. It is considered the primary mediator or the main mediator of neocortical information that is going to the hippocampal formation. So the neocortical information that entering the hippocampal formation pass through the entorhinal cortex. It's the main mediator. Entorhinal cortex has connections with with olfactory pathways. As I mentioned earlier, that olfactory pathways connect with the extractive limbic system, and entorhinal cortex is one of them. And therefore could form part of the piriform lobe, which is considered the primary olfactory area. We think that entorhinal cortex could be also a big region with some zones. And so there are zones which have this olfactory role, and then there are some zones which actually have the limbic functions. This image shows you that anatomy. So this is basically the piriform cortex, which is considered part of the primary olfactory area. And in that piriform cortex, we can see the entorhinal cortex overlapping. Well, the other components of the piriform cortex or the piriform lobe will be the ancus of the parahippocampus and the lateral olfactory stria. In coronal section, the entorhinal cortex is just beneath the other components of the hippocampal formation, as I mentioned earlier. So in coronal section, so this is a left coronal section through the temporal lobe for us to see the components of hippocampal formation. We can see entorhinal cortex there, lower than the other components of the formation. One important thing that we know about entorhinal cortex, commonly called area number 28, is that it gives rise to some efferent pathway that goes through the subiculum to reach the dentate. This pathway is known as the perforant pathway. We call it perforant because they go through subiculum. Remember that this information is, maybe it came from the neocortex and now passing through entorhinal cortex, which processes it, then relays information to the dentate gyrus and perhaps hippocampus. So we call that the perforant pathway. It's an important root, and that's what I was saying, that internal cortex forms a main root. It's the main connection between the neocortex and hippocampal formation. We can see some projections there to the dentate region. We can also see some projections which we think can also go to the hippocampus without necessarily going to the dentate region. All right, so that is entorhinal cortex. The other component of the formation, the subicular complex. So if you follow again, this is parahippocampus, and so this is part of entorhinal cortex. Follow the cortical material, this is the subicular region. If you follow that further, this is the hippocampal region, and that's the dentate, the last component. So we are now talking about the subicular region. The subicular region, so this is also showing us that the entorhinal cortex is somewhere here, then subicular, then hippocampal fields, and lastly, the dentate. The subicular complex is a transitional zone between the 6-layered entorhinal cortex. Entorhinal cortex is 6-layered, although the 6 histological layers of the entorhinal cortex may not necessarily be directly equated with naming in terms of what each layer has with the 6 layers of the classical neocortex. However, the entorhinal cortex still has 6 histological layers. They may have different architecture. So moving from this, the subiculum doesn't have these 6 layers. It actually has 4 to 5 layers. So it's a transition zone between the entorhinal cortex and the archicortex, which you're going to see in the hippocampus and dentate gyrus. The components of the subicular complex are subiculum, presubiculum, and parasubiculum in terms of where they are found. So from the entorhinal cortex, we first move to the parasubiculum, then presubiculum, before we finally reach subiculum. We know that subiculum is also important in giving efferents from the hippocampal formation. The efferents that subicular give tend to be subcortical, or they're having subcortical termination. So they're not necessarily landing onto cortices, but they land into nuclei specifically, and especially the septal nuclei. So this image shows you the regions we call the entorhinal cortex, giving you fibers, the perforant pathway, which goes to the dentate as well as to the hippocampus in blue, but also shows you the fibers which are in the subicular area in green, arising from there and going around the hippocampus and traveling through a particular tract, which you're going to name later as the fornix. So in simple terms, subiculum is one of the areas that give rise to efferents from hippocampal formation. Third component of the hippocampal formation is the hippocampus itself. You may want to call it hippocampal gyrus or just hippocampus or other terminologies. It lies above subiculum, as we just seen, and medial to the parahippocampal gyrus. When you look at it from above, it forms an elevation along the floor of the inferior horn of the lateral ventricle, as we can see in this particular image. So this is the hippocampus. Anteriorly, it has some things which look like knuckles, and it's a bit swollen anteriorly than posteriorly. In this particular image, it's a coronal image of the left temporal lobe. Mesotemporal lobe, we can see the hippocampus is above, but the subiculum is below and entorhinal cortex much lower. Histologically, the hippocampus has 3 layers. So it is an archicortex. So having the outer molecular layer, a mid layer that is pyramidal, and an inner layer that is polymorphic, having a mixture of gray and white matter component. However, in terms of how these three layers are organized histologically, we have some 4 fields which have been described. So you can see CA1, CA2, CA3, and CA4. Though CA4 is considered the core of the dentate. I'll not go into the histostructure of this particular hippocampal fields, but just understand that in terms of cytoarchitecture, the arrangement of the 3 layers here in terms of the structure of the cells and the population, the number of the cells are a bit different from this zone and a bit different from that zone and perhaps that zone. In terms of functions, hippocampus is important for 2 major things. There are many other associations that can be put, but let's consolidate 2 here. Hippocampus is known to consolidate information that is important for transfer of memory from short-term to long-term memory. So it's very important in consolidating information from short-term memory to long-term memory. Very important. So that means it's important in learning. Another key function of the hippocampus is that's important in spatial navigation, navigating your environment. So basically important in finding where you are, where the body is in the environment. No wonder some of the disorders of the hippocampus will be related to memory loss and perhaps even with epileptic disorders. The major efferent fiber from the hippocampus and indeed hippocampal formation is the fornix. It's a big structure. So this is the hippocampus. And on top of it, that thin structure is the fornix that comes to become large. So this is the fornix. Is the main efferent from the hippocampus and indeed from the hippocampal formation. Disorders of the hippocampus are associated with clinical conditions such as amnesia, Alzheimer's disease, epilepsy, and schizophrenia. We'll talk about a few of these. The 4th component of the formation, the dentate gyrus. So remember that if you follow this, this is the entorhinal cortex, subiculum, hippocampus, then dentate. The dentate gyrus is a crenated stripe of cortex. So it's also a 3-layered cortex located between the hippocampus above and subiculum below. In this particular image, you can still see that. So subiculum below, hippocampus above. So this is the dentate gyrus. And in this image, we can see what you are referring to as the crenation, those convolutions that its surface has. This is the dentate gyrus. This is the hippocampus itself. This is part of the fornix arising from the hippocampus. And this region here is the subicula, which is, if you are to extend it further, then we'll be talking about entorhinal cortex here. What we know about the dentate gyrus, one of the things we know is that it's one of the most plastic regions in the brain, exemplified by its ability for postnatal neurogenesis. This is a very rare thing in the brain for neurons to generate after birth. However, the neurons of the dentate gyrus are known for capacity for postnatal neurogenesis. Perhaps that is linked with its key functions. It is important in contributing to the formation of new episodic memories. So these are new memories which are different from former ones. And they have been linked about, that's why perhaps you need new neurons to come up to form the new memories. Maybe it's a theory. It's also important in spontaneous exploration of some novel environment, something you've never done before, but you're just exploring some new environment, so to register new memories. That's dentate gyrus. Great, so we've looked at the 4 major components of the hippocampal formation. But as I mentioned to you, we also have what we call hippocampal rudiments. And so I want us to go through the hippocampal rudiments. So these are vestigial extensions of the components of the hippocampal formation beyond the temporal lobe. So there are extensions out, which are found out of the temporal lobe. There are 3 of them. We'll talk about them. This is what we call indusium griseum. Now follow with me from dentate gyrus backwards. So this is dentate gyrus. Following back, we can see that dentate gyrus becomes what we call the fasciolar gyrus. So this is where the dentation ends, but it's the same structure. The gyrus fasciolaris is just below and behind, and essentially below the splenium of the corpus callosum. And then extending from there is what we are calling the indusium griseum. So this is actually a gray matter that lies above or on the dorsum of the corpus callosum, and we can trace it to the dentate gyrus through gyrus fasciolaris. Because it lies above the corpus callosum, we can also call the supracallosal gyrus. In this particular image, you can see the same thing. So that's the supracallosal gyrus or the indusium griseum. And follow with me how it comes on the middle aspect. So this is a genu of the corpus callosum, that's the rostrum corpus callosum. And now we see 2 things here. We see the parolfactory area, and we see the gyrus subcallosus, or basically the subcallosal gyrus, which is the second component of the hippocampal formation. Sorry, hippocampal rudiments. So subcallosal gyrus is also known as the paraterminal gyrus. It's a narrow stripe of extension of the indusium griseum on the middle aspect, in front of what we call the anterior commissure, this one, lamina terminalis, this one, and behind the parolfactory area, or what we had called earlier the subcallosal area. Remember, I mentioned to you that there's some conflict of terminologies, and so we can settle on this one in this particular discussion, that the parolfactory area is also called the subcallosal area, and it is different from what we're calling the subcallosal gyrus, which is also called paraterminal gyrus. Okay. In this particular image, we can still follow that. So that's the indusium griseum, gray matter above corpus callosum. Following it anteriorly, we can see that, and so we go back that narrow stripe. So that's the subcallosal paraterminal gyrus. Anterior to that is the subcallosal area, and behind that is the anterior commissure and lamina terminalis. There's evidence that this one will also continue into something here, which we call the amygdala, through another connection there that we call the diagonal band of Broca. Some people may consider it part of the rudiments. I don't particularly do that, and so I'll not do it here. Okay, the third component is what we call the medial and the lateral longitudinal stria. So we have what we call longitudinal stria, which are just fiber bands running on either side of the indusium griseum on the dorsal surface of the corpus callosum. So these are the fiber bands. So you can see they run longitudinally. So this has been dissected to show the fibers of the corpus callosum as they run transversely. In this image, remember the commissure of fibers. But the longitudinal stria are anteroposterior fibers, so to speak, so they're not transverse. So this is the lateral stria. The medial stria is here. The right and the left medial stria are most likely very close together in this particular region because this is the lateral striatum on the other side. Okay, so that summarizes the components of the limbic lobe. We've looked at the limbic cortex. We've also looked at the hippocampal formation. We can now put our attention to the next component of the limbic system, which are the nuclei. So remember, there are subcortical gray matter aggregations which are associated with the limbic lobe. We can list them here and then talk about each of them. All right. Let's start with what we call amygdala nuclear complex. It's located there. We also have what we call the mammilla nucleus, which is that one. We have the anterior thalamic nucleus there. We have the septal nucleus somewhere there. We have what we call the ventral striatum, not shown in this particular image, but includes something called the nucleus accumbens. And lastly, we have the habenula nucleus, which is somewhere here. I want us to begin our discussion with the amygdala nuclear complex. So this is the location of the amygdala nuclear complex. This is still a coronal section through the left hemisphere, but it's anterior to the location of the hippocampal formation. So we are not seeing the folds of the hippocampal formation, but in front of that formation, if you take a cut, you'll see now the amygdala nuclear complex. It's an almond-shaped cluster of nucleus. Indeed, there are several nuclei. Can break them down into some number. You might get even over 10 nuclei within the amygdala nuclear complex. But they have been grouped in different ways. I'll not go into the grouping terminology because, again, that will bring a lot of confusion. So let's just keep it simple. This cluster of nuclei is located in the temporal lobe and in particular anterior to the hippocampus and beneath what we call the uncus. So if you are to look at this image, this is the hippocampus. Now the nucleus is located anterior to it. So that's why if you take a coronal section anterior to the hippocampus, you'll see the nucleus, but you don't see the formation. If you take a coronal section through here, then you will see the formation and you will not see the amygdala nuclear complex. Important to note is that this cluster of nuclei receives input from various sensations, whether somatic or visceral. There's no distinction. It actually receives from multiple sensations, from vision, hearing, somatic sensations like pain, pressure, position sense, olfactory system, and taste system. They project information to the amygdala. So here you can see different nuclear group. Again, I'm avoiding the terminology so that we are not mixed into many things, but remember there are many nuclei and perhaps one of them is just designated for receiving those sensations, although there are some internal circuits. And from there, you'll have output information that also goes to various regions of the brain. In terms of how the sensations enter the amygdala, perhaps it's important to highlight. So the stimulus will come, and we know that sensations usually pass through thalamus. So they pass through thalamus as ascending tracts. Maybe these are second-order neurons. They terminate in the thalamus. After terminating the thalamus, we expect the third-order neurons to go to the cerebral cortex for perception, and indeed that happens. But we can also have fibers arising from the thalamus to the amygdala nuclear complex direct. So that's one way in which amygdala receives sensory input from the thalamus direct. However, this was another route. Once the sensory information has reached the primary sensory cortices and perhaps sensory cortices in general, there's some connection between the sensory cortices and amygdala as well. So that second route through which it receives the sensory information. But yes, there's another one. From the cortex, from the neocortex, information can go to the hippocampus. Maybe through cingulate, then perihippocampus, then through entorhinal cortex to the hippocampus. This information from the hippocampus can also go to the amygdala. So we have connections. Sensations reaching amygdala from hippocampus, sensation reaching amygdala from the cortex, and sensation reaching amygdala from the thalamus. There are many nuclei within the amygdala that process those sensory inputs. These nuclei also have multiple connections with different parts of the central nervous system. And just look at that chart and you'll see what I'm saying, that we have amygdala having connection. Almost every part of the central nervous system has connection to the cerebral cortex, as you can see. It has connections with the olfactory areas. It has connections with the hippocampus. It has connections with the brainstem. So these are connections to the brainstem, basically. It has connections with the basal ganglia. It has connections with the hypothalamus. It has connections with the thalamus. This is still basal ganglia as well. So amygdala connects with many things around it. This image tries to just narrow down to which nucleus of amygdala connects with which part. Again, it's a very complex story. So you can just keep it simple that yes, the amygdala nuclei have connections with many parts. Maybe we can try to now mention examples of just amygdala nuclei. So you can see lateral amygdala there, that are big nucleus. You can see the basolateral amygdala there, a big nucleus. Basomedial there, we have the central amygdala, the cortical nucleus of amygdala. So there are many amygdaloid nuclei. And in these nuclei, you'll find that there are even subgroups of nuclei, like in the basolateral amygdala, there could be some subgroups of amygdala. And as I told you, that if you have to break them down into details, you may have even over 10 different types of nuclei within the amygdala nuclear complex. Let's make it this way. It has extensive connections with several parts of the brain: hypothalamus, thalamus, the cerebral cortex, some subcortical nuclei, quite extensive connections. So what are the key functions of amygdala? It is known that amygdala is important in fear conditioning and aggression responses. So it's the one that conditions you in the management of fear and developing those fear responses. You may choose to respond by being aggressive. If something that's threatening comes to you, or you may choose to not do anything. But generally, the usual response that when something that is intimidating comes, you have some fear response, and you may choose to protect yourself by aggression. Amygdala is important in that way. It is also important in helping us to tie emotional meaning to memories, reward processing, and decision making. So it helps us to link emotion to this thing. When you have emotional meaning to memories, they tend to last long, we understand, compared to if particular memory didn't have any consequential impact in terms of emotion. It could be positive or negative emotion, doesn't matter. And so this is important. There are many other functions which have been linked with the amygdala. For example, sexual orientation, that's a big debate. Behavior, how you choose to behave sexually, anxiety, it has a link with addictions, and the like. So there are many key functions, or there are many functions which have been associated with the amygdala nuclear complex. The other subcortical nucleus to talk about is hypothalamus. We'll talk about hypothalamus in the context of the limbic system. Hypothalamus has several functions because it has several nuclei within it. It forms part of the lateral wall of the third ventricle, and it's located just below the thalamus. The sulcus separating thalamus and hypothalamus is this region, which we call the hypothalamic sulcus. So there are several nuclei within the hypothalamus, and this image tries to just give us a glimpse of what that really means in terms of the hypothalamic nuclei. These nuclei have several functions. Some functions may overlap, but there are several functions. Even categorization of hypothalamic nuclei is a very big, complex thing, whether you want to look at them from anterior to posterior or medial to lateral or above downwards, there are many nuclei. So these nuclei have several functions, which we're not focusing on in this particular lecture on the limbic system. But also important to note is that these hypothalamic nuclei have connections with structure of the limbic system. And important, they also have connections even with the amygdala itself. So this image shows you hypothalamic connections with the amygdala, as you can see. So this is the amygdala. The green represents a path, which we call stria terminalis. And these neurons actually terminate within various nuclei within the hypothalamus. We can also see that hypothalamus has connections with the hippocampal formation, now the red. This is now through the fornix, which the fibers also terminate within hypothalamus, and importantly, in what we call the mammillary bodies of hypothalamus. Well, the septal area, we're going to talk about septal nucleus shortly, but we can also see connections in septal area and hypothalamus. So hypothalamic nuclei connect with various components of the limbic system. That's the message. The mammillary nucleus itself, it's an important one as well, because it forms part of a key pathway we are going to talk about. It's located in the inferior aspect of the hypothalamus, and it's actually visible from the base of the brain. In this particular image, we can see the shaded, the highlighted ones. Those are the mammillary bodies of hypothalamus. Each mammillary body has two nuclei. So we have the medial lateral mammillary nucleus, located within this region, which we call the interpeduncular fossa, and the mammillary bodies. Now, we know that these mammillary bodies, they contain the mammillary nuclei, and these mammillary nuclei form part of connections which are important in the limbic system. And we're going to talk about what we call the Papez circuit. It's an important part. It receives afferents from the fornix. So remember, fornix is a major efferent from the hippocampal formation. So it receives afferent from the fornix and give efferents which go to the thalamus, particularly through this route, which we call the mammillothalamic tract. May also give you efferents which go to the tegmentum, the brainstem. You may want to call that one mammilotegmental pathway. Okay, I've already mentioned to you that it receives input from the fornix, via fornix, and this input via fornix is an output from the hippocampal formation. So this image shows you, we can follow this one. So this is the mammillary body of hypothalamus. This is fornix, and we can trace that one all the way to the hippocampal formation. All right, I hope you've seen that. The other nucleus talk about is the anterior group of thalamic nuclei. So this is an axial section of the brain's an MRI image, what we call T2 MRI, because fluid is bright. So this is the caudate nucleus, its head in particular. This is lentiform. Those two are part of the basal ganglia. Posterior and medial to that is this large mass of nucleus, which is the thalamus. So you can see we have two aggregations. So we have a right and a left thalamus. The thalamus is part of the encephalon, and it's actually a large mass of nuclei on either side of the third ventricle, which is this slate. If you are to draw them, they may look like this. So the large masses, the left and the right one. The anterior thalamic nuclei, the ones shaded yellow, are actually part of the many nuclei that thalamus has. It's an enclosed region, enclosed by the two arms of the internal medullary lamina, and lamina that separates the thalamus into three nuclear groups: anterior nuclear group, the medial nuclear group, and the lateral nuclear group. This medial nuclear group is the one that will have what you call the medial dorsal nucleus. The lateral nuclear group has many nuclei, others in the dorsal aspect, others in the ventral aspect. Our main intention talk about the anterior nuclear group, the yellow. So the anterior nuclear group consists of three nuclei. We have an anteroventral, which is on the lower side, anteromedial, which should be on that side, and anterodorsal, which should be on this side. These three nuclei of three nuclei of the thalamus are the ones that are linked to the limbic system. They primarily receive information from the mammillary bodies of the hypothalamus through what I just mentioned in the previous slide as the mammillothalamic tract. In this image, we can see that understanding. So from the hippocampal formation, we have the fornix going round there. And then fornix lands to the mammillary bodies of hypothalamus. From the mammillary bodies of hypothalamus, we have an efferent to the anterior thalamic nucleus through this mammillothalamic tract. The next nuclear group are the septal nuclei. The septal nuclei are on the septum. The septum consists of the upper part, what we call the septum pellucidum, and the lower part, what we call the septal area. So this is septum pellucidum, and this is septal area. In coronal section, so if this is the corpus callosum, this is the septum pellucidum, and the lower part is the septal area. The septal area has about 4 nuclei within it that constitute what we call the septal nucleus. So we have nuclei within this area. We call them septal nuclei. This septal area in gross anatomy will be somewhere around there, as I've just shown you. So that's the septal area. We can see that it has various connections. It receives information from multiple places. You can see information arising from amygdala, going there. You can see information arising from hippocampal formation through fornix, may extend there as well. So it has multiple connections. In this image, you can see that. So from amygdala, the blue, that goes to the septal area. That is called the stria terminalis, from the amygdala to the septal area. It doesn't just go to septal area only, it will also go to maybe hypothalamic regions, but we are focusing on the septal nucleus now. Even the phonics, we'd already mentioned the projection to the mammillary nucleus, but we can also see projection to the septal area as well. Now, it receives information from multiple places, as we just mentioned. Even the olfactory area, you can see projecting the septal area. It has connections again to multiple regions. Maybe we just pick two efferents from it. One to the habenula nuclei. We can see that path. We'll give it a name when we talk about the tracts. We can also see this one from the septal region to the hypothalamus. Again, we give it a name when you look at the tracts. Great. So that was the septal nuclei. Now let's talk about the ventral striatum. The ventral striatum, which is also an important region in the limbic system, simply means the ventral aspect of the basal ganglia. This ventral aspect of the basal ganglia is likely a confluence of two major nuclei of the basal ganglia. This one, the caudate nucleus, especially the head of caudate nucleus there. And this one, the putamen. So the point of union of the caudate nucleus and the putamen in this shaded region, which are called the ventral striatum. Ventral striatum consists of two major nuclei within it, the nucleus accumbens and the olfactory tubercle, olfactory tubercle being much lower. This ventral striatum has connections with several parts of the brain, including the cerebral cortex, the hippocampal formation, the amygdala, hypothalamus, and the brainstem. And this image tries to capture for us those connections. So let's consider the whole of this to be the whole striatum or basal ganglia, so to speak. We have the dorsal aspect of the striatum and the ventral aspect of the striatum. The dorsal aspect of the striatum is the one involved with some movement functions. So it has connections with the cerebral cortex. So these ones are largely cortical, the cerebral cortex, the anterior aspect. And it has also connections with some parts of the brainstem. Okay, so this one is largely for movement functions. Then the ventral aspect of it, the ventral striatum, which has now the nucleus accumbens, has also connections with many parts, including now parts of the limbic system. We can see connection with the insula. We can see connection with the amygdala, connections with the hippocampus. So connections with the hypothalamus. There are many connections of the ventral striatum with components of the limbic system. We know that nucleus accumbens is involved in acquisition and development of reward-based behaviors. And this is also tied with the drug addiction and the drug-seeking behaviors. If you go through an experience that looks like it's exciting, making you feel nice, you'd want to go through it again and again. And so addictive behaviors have been associated with activities of the nucleus accumbens. Last but not least is the habenula nuclei. Habenula nuclei are two nuclei, the medial and lateral habenula nuclei, that are bilaterally located within the epithalamus, this region there, just in front of the pineal body. Pineal body is that one. So this is habenula nuclei. These also have extensive connections with multiple parts of the central nervous system and are therefore maybe having multiple functions as well. In this image, we can see connection with the septal area, perhaps through that yellow structure known as the stria medullaris thalami. Others have not been shown here. But in this particular image, we can see extensive connections of the medial and the lateral amygdala, medial lateral habenula nuclei with multiple parts of the brain. The green represents afferents to the habenula nuclei, the blue represents efferents. So in a nutshell, we can say it's connected to many things: pineal gland, the basal forebrain, the medial prefrontal cortex, hypothalamus, basal ganglion factory, all these provide input to the habenula area. And then it has connections which are efferents, go outside and essentially targeting the brainstem structures in particular. In many words, habenula nuclei are important in regulation of monoamine neurotransmitters, and especially regulation of dopaminergic activity and regulation of serotonergic activity. So largely control these monoamines, and those are the ones you see here in relation to the brainstem structures. When you look at the major connections to the habenula area, the main fiber afferent is the stria medullaris, which you can call stria medullaris thalami to distinguish something else in the fourth ventricle. Stria medullaris is a major afferent to the habenula area, and the major efferent from it is this one, what we call fasciculus retroflexus, which is an output tract which targets structures in the brainstem, especially in the midbrain region, as you can see there. Great. So we have successfully covered the second major component of the limbic system. These are the nuclei of the limbic system, the nuclei associate the limbic system. So the last component that we need to cover are the fiber tracts of the limbic system. Remember that these are neuronal pathways that connect various nuclei and cortices in the limbic system, and they're equally important. There are quite many. I'll give you a list of them. Then we'll discuss a few. So we have what we call phonics. This is the phonics. We have cingulum located within this region, cingulate gyrus. We have stria terminalis, the blue. We have stria medullaris thalami, the yellow. We have mammillothalamic tract, this one. So those ones in green are relatively large, and it's possible to see them. You are able to see them easily from gross anatomy specimens. I'll give you still other list. So we have the fasciculus retroflexus, not shown in this particular image. We have the perforant pathway, again not shown in this image. The ventral amygdalofigo pathway, the diagonal band of Broca, I think it's shown there. And the medial forebrain bundle, maybe not shown here, but could be part of this screen there. So the ones in blue are a bit difficult to see from gross anatomy specimens. If you're doing a practical, you may not want to waste a lot of your time looking for them. The ones in green, you can easily see from gross anatomy specimens. If you're doing a practical in neuroanatomy, you can easily see them. I want us to discuss them one by one. Let's start with the phonics. So phonics is this one. It's a large fiber bundle that houses efferents that arise from the hippocampal formation. This image, you've already seen it. So this is the hippocampus. So this is the phonics containing efferents from the hippocampal formation. Anatomically, we describe the phonics as having 5 parts. So we have this region here, which we call the fimbria of phonics. Then we have this region suspended beyond the hippocampus, which we call the crus of phonics. When the right and the left crus are almost meeting, not yet met, there's some fibers that cross to join the two, and that constitute the phonics commissure, or others known as the hippocampal commissure. Then beyond the commissure is the region where they have now met, they're in contact, right and left. That's what we call the body of phonics. And then finally, we have anterior and posterior columns of phonics with reference to the anterior commissure, this anterior commissure. So these are part of phonics, a bit little, but yes, there's some fibers that go in front of the anterior commissure. We call them anterior column. That those fibers which are behind the anterior commissure, we call them the posterior column of phonics. Sometimes we use the term phonics, but use the term pre-commissural and post-commissural fibers. The post-commissural fibers are basically the posterior column fibers. The pre-commissural fibers are the anterior column fibers. The commissural term is with reference to the location of the anterior commissure. This image shows you a better outline anatomy of the phonics. So the fimbria attached, it's a membrane attached to the hippocampus, then the crus, then the commissure, or so you can call it the commissure of phonics, or you can call it hippocampal commissure. Then you have the body of phonics and the columns of phonics. The ones shown here are both posterior columns because they go to mammillary bodies, the right and left mammillary bodies. So the anterior columns are not being shown in this image, but in this other image, you can see the anterior columns and the posterior columns of phonics. The anterior columns are the precommissural fibers. The posterior column are the postcommissural fibers. The phonics connect the hippocampal formation to the mammillary and septal nucleus. So the anterior column will largely be going to the septal area, posterior column to the mammillary nucleus of hypothalamus, and they come from hippocampal formation. This image shows you the extension to the septal area through the anterior column and extension to the mammillary nucleus through the posterior column. The second tract is the cingulum fibers. The cingulum fibers are long curved association fibers found within the cingulate gyrus as well as the parahippocampal gyrus. So these are diagnostic images showing us the cingulum fibers. What we note from these images is that the cingulum is not a unitary pathway, as we can see some fibers joining and leaving, but multiple fibers. However, within this tract, there are those long fibers which may go all around, but most of the fibers do not quite do that. They join and leave at some point. So it contains fibers that connect cingulate gyrus with other regions of the cerebral cortex, and also, of course, merging into the parahippocampus. So this image shows you the fibers joining and leaving, but they all constitute the cingulum, with others actually going round. The ones that go round are actually headed to the parahippocampal gyrus, within the parahippocampal gyrus. The functions of the cingulum are strongly linked to emotions, social interactions, motivation, attention, pain, and memory. We've said almost everything because cingulum fibers connect multiple things. Straterminalis is the second fiber tract, the third fiber tract to talk about. Straterminalis, the blue, it's an output tract from the amygdala nuclear complex to the hypothalamus and septal area. We can see it here. So this is the amygdala. So the blue is the stria terminalis, going to the septal area and hypothalamus. The green is the one we are referring to in this particular image, again, efferent from amygdala to the hypothalamus as well as the septal area. Usually the stria terminalis will pass between the caudate nucleus and the thalamus within a groove. We call that groove the caudothalamic groove. So maybe we're not seeing the caudate nicely there. But this is a gross anatomy image. We can see the caudate nucleus there, and this is thalamus. So this is the stria terminalis. In this image, this is thalamus and this is caudate, the body. So stria terminalis is located in this gap, or in that where the dots are. Usually the stria terminalis houses a nucleus, which we call the bed nucleus of the stria terminalis. It's considered a continuum of the amygdala, really. So there's some gray matter within the stria terminalis, but generally considered a continuum of the amygdala. That there's some nuclear aggregations along it, which is basically a continuum of the amygdala. There's some terminologies about that, which I really don't want us to go through in this particular class. All right, so we have talked about the stria terminalis. The other fiber tract to talk about is the stria medullaris thalami, now the yellow one. Stria medullaris thalami forms a ridge on the middle aspect of the thalamus. This is the thalamus. And in this image, this is the stria medullaris. Now, there's another structure called stria medullaris in the floor of the fourth ventricle. To distinguish the two, most authorities prefer to call this one stria medullaris thalami. That said, not everyone actually does that. So some people prefer to just still call it stria medullaris around the thalamus and stria medullaris of the fourth ventricle. So you just make your own decision, but understand that there's two concepts there. Stria medullaris thalami is that one. It contains afferent fibers which go to the habenula nuclei. These fibers come from multiple places, but especially from the hypothalamus and perhaps from the septal area as well. The fibers within the stria medullaris may go beyond habenula to also influence the functions of the pineal gland. The cell types within the pineal gland are called pinealocytes. They produce a hormone called melatonin, which is important in controlling the day-night rhythm. And so through that, we can say that even these fibers are important in controlling the circadian rhythm, the day-night rhythm. All right, the other fiber tract talk about the mammillothalamic tract arising from the mammillary nuclei within the mammillary bodies of the hypothalamus and terminating into the anterior thalamic nucleus. So this is the mammillary nucleus. And we can see connection between that and the anterior thalamic nucleus. This is what we call the mammillothalamic tract. In this image, we can see that gross anatomy dissection, very meticulous dissection to show the tract. So this one from here, this is the mammillary body. We can see joining it is actually the posterior column of fornix, or what we call the post-commissural fibers of fornix. So this is fornix landing to the mammillary body. Then from there we have the mammillothalamic tract. So this mammillothalamic tract, that is the stria medullaris that was going to the habenula area. And we can see something coming from there down the habenula area, the fasciculus retroflexus. It's another tract that I already mentioned. Okay, let's begin there. So what are the other fibers apart from the 5 I've already given, which are easily seen. We have others that are a bit difficult to see, but I'll still mention them. Fasciculus retroflexus, which is also called habenular interpeduncular tract, contain fibers from the habenular area to the interpeduncular fossa, to the interpeduncular area within the midbrain. The perforant pathway, I already mentioned it. It contains fibers from the entorhinal cortex. They go through the subiculum and terminate within the dentate gyrus, as well as some fields of the hippocampus. The ventral amygdalofugal pathway, as the name suggests, arise from the amygdala, but they pass in front as opposed to passing behind. Remember, the ones that pass behind were the ones which are in the strata terminalis. So this yellow structure is the amygdala. There's 2 tracts from it, 2 major efferents from the amygdala. The one that's going dorsally constitute the stria terminalis, and the one that passes anteriorly. So the one that's passing anteriorly is the ventral amygdalofugal pathway. We can see it connects the amygdala to multiple areas: olfactory regions, septal area, hypothalamus. In this image, we can see amygdala there. The dorsal root is the strata terminalis in blue, and the ventral root, the ventral amygdalofugal pathway, is what you expect there. Then we have the diagonal band of Broca. The diagonal band of Broca interconnects amygdala to the septal area. Now, the diagonal band of Broca is considered, or is considered, maybe considered by some authorities as part of the rudiments, because we can see lower extension from the paraterminal gyrus to the diagonal band of Broca. Many authorities do not consider it part of the hippocampal rudiments, and some also not considered there. Last of the fibers is the medial forebrain bundle. The medial forebrain bundle is an important fiber tract that connects the limbic forebrain to the brainstem structures. So the limbic forebrain, consisting of perhaps the septal area and the hypothalamus, the fibers arise from there and project the brainstem, and there are many structures in the brainstem that receive the medial forebrain bundle. So the ones in blue here constitute part of that tract which are on the medial forebrain bundle, but goes beyond that. And in this region, we can see the fibers that constitute medial forebrain bundle arise from the septal nuclei. They also arise from nucleus accumbens, and indeed they usually also arise from the hypothalamus, and they project to the brainstem as part of the medial forebrain bundle. Something related to that is what we call the dorsal longitudinal fasciculus, which also carry information down from the hypothalamus to the brainstem, and these two are important in modulating limbic and autonomic functions down to the brainstem, and that's why you see them going to what we call the reticular formation, which has limbic as well as autonomic functions. Great. So those are the major components of the limbic system. We've taken quite a long time there, but we've gone through details of the components of the limbic system. Now I want us to look at briefly the circuits within the limbic system and some disorders. So there are 3 major circuits I'll talk about. That does not mean that there are only 3. There are many, but I'll pick 3 which are commonly talked about. There's what we call the intrinsic circuit within the hippocampal formation. So this is a circuit that is intrinsic to the hippocampal formation. It's a circuit that revolves around 4 structures: entorhinal cortex, So fibers from entorhinal cortex project to the dentate gyrus through the perforant pathway. From the dentate gyrus, we have fibers projecting to the hippocampus through what we call the Morse fibers. So the hippocampus is what we call the Ammons horn or Conu ammonis, and that's why these ones CA, Conu ammonis 1, Conu ammonis 3. Let's just call this one the hippocampus and forget about this confusion to keep it simple. Then from the hippocampus, we have fibers connected to the subicular complex, and from subicular fibers going back to the entorhinal cortex. So if you remember, the fold still structure very close to one another. And so it's just an intrinsic circuit. It's a circuit intrinsic to the components of the hippocampal formation. It is believed that this is the circuit that underlies functions of short-term memory. The information just revolves around there, revolves around there, revolves around there over some time. If enough potentiation occurs or reinforcement occurs, it may then be transferred to long-term memory, and hippocampus will be vital in that conversion. And that's why we said the hippocampus is important in conversion of short-term to long-term memory. It's therefore important in learning. Therefore, the intrinsic circuit is still important in learning. The second circuit, which is very famous for the limbic system, the Papez circuit. This circuit involves a number of structures. So we have the cingulum, which are fibers within the cingulate gyrus. Those fibers project to the parahippocampus. And so within the parahippocampus, we have projection to the, or extension to the entorhinal cortex. Then from the entorhinal cortex, we have projection to the hippocampus through the perforant pathway, of course, now dentate to be somewhere in the middle here. From the hippocampus, the major efferent from the hippocampus, the fornix, and fornix through its post-commissural fibers project fibers to the information to the mammillary bodies, so therefore through fornix. From the mammillary bodies, we have tracts going to the anterior thalamic nucleus through the mammillothalamic tract. And then from the anterior thalamic nucleus, we have projections again to the cortex, including now cingulate cortex, through what we call the internal capsule, a projection fiber connecting thalamus to the cerebral cortex. We've made a complete round, and that complete round is what we call the Papez circuit. We can do it here again. We can start from the cingulate cortex. So from the cingulate cortex, we have cingulum fibers that travel, and let's say they converge through the parahippocampus. Then from the parahippocampus, at around the region of the entorhinal cortex, we have information going to the hippocampus through the perforant pathway, so that will pass through dentate. Then from the hippocampus, we have efferent information leaving hippocampus through the fornix, going all the way, and because you're following Papez circuit, we'll take the posterior column of fornix to the mammillary bodies. From the mammillary bodies, we take this route to the anterior thalamic nucleus, so this is the mammillothalamic pathway. And then from the anterior thalamic nuclei, we have fibers coming from there to the cerebral cortex through the internal capsule. We've gone back to where we started. So this is a Papez circuit, the circuit that was described by Papez, and he believed that this is the circuit that underlies emotions. Then we have the basolateral circuit. The basolateral circuit involves the basolateral nuclei of amygdala. So it involves the thalamus. And in this particular time, we're not talking about the anterior thalamic nuclei per se, but the dorsomedial nucleus of thalamus is the one that's involved here. So we have the dorsomedial thalamic nuclei, which has connections with orbitofrontal cortex. Remember, this part of the limbic cortex. Then orbitofrontal cortex projects to the temporal cortex through what we call the uncinate fasciculus. The uncinate fasciculus is an association fiber that connect the frontal lobe to the temporal lobe. So that is the uncinate fasciculus. It's an example of an association fiber, long association fibers. So from the anterior temporal cortex, we have information reaching the amygdala. Remember that amygdala is actually located in the anterior temporal lobe. So the nucleus of amygdala involved here is the basolateral amygdala. Then from the basolateral amygdala, we have information again going through the ventral amygdalofugal pathway to reach the thalamus. Remember, there are two major efferent fibers from the amygdala, the strata terminalis and the ventral amygdalofugal pathway, which goes to multiple places, including thalamus. They're also going to the septal area. So for the basolateral circuit, this is the circuit. This circuit is believed to encode information about social signals and social plans for social acts. So for example, if somebody's talking to you and there's a particular way they are showing their facial expression, you are able to encode some information from there, even if they're not saying it. So for example, when somebody's winking, you know what that means. When they frown, you know what that means. And this is why it's linked with the human ability to infer intentions of others from their language, from their gaze, from their gestures. Sometimes when someone is talking to you, you are able to pick from their tone what they actually intend to do, even if they're not saying it. Or when they look at you, you're able to infer what they're intending to do, even without them saying it. So this is a system that helps us to code that. All right, so those are the major circuits of the limbic system. We will now finish the long story by talking about some clinical aspects of the limbic system, and we'll start with the mesial-temporal lobe epilepsy. This is the most common type of epilepsy disorder, and especially the focal epilepsy disorders in adults. Its commonest cause will be atrophy or sclerosis of the various components of the hippocampal formations. We see atrophy involving the dentate gyrus, the entorhinal cortex, and the hippocampus there. Perhaps you're wondering how I picked that. So let's compare the volume of hippocampal formation on this particular side. This is the left side of the patient, and this particular side, the right side of the patient. You'll see that this is a bit full compared to that side. Remember, we expect some symmetry, but this is a bit atrophied, small, compared to this side, which is relatively full. We can try also to see another image. So compare this side and that side. This is relatively small compared to this side, which is relatively full. So in addition to the various components of the hippocampal formation, amygdala is also involved in this particular lesion of which we're talking about mesial-temporal lobe sclerosis. Amygdala is also involved. So there's atrophy of the amygdala as well. This is a recording. I want to play it so that you see the difference between the right hippocampal formation volume and the left hippocampal formation volume. So we are scanning through We're going posterior now. That's why you're seeing cerebellum, but you're seeing components hippocampus. So compare this side with that side. You notice that this one is smaller compared to this side, which is relatively full. We've now scanned onto the anterior aspect. We're going back again. There we are. So this side is full compared to that side. This was obtained from a patient who had mesiotemporal lobe epilepsy. The other syndrome to talk about is what we call Kluver-Bucy syndrome. Kluver-Bucy syndrome is caused by bilateral distraction of amygdala. So you involve the right and the left amygdala. The clinical presentation of this disorder include the following. So we have docility or placidity. So this is basically loss of normal fear and anger responses. The people affected present with low aggression, and they have diminished fear responses. They also have hyperorality, which is a compulsive tendency to put things into the mouth. They don't have to be food. It's just anything into the mouth, even stones. Then there's hyperphagia. Hyperphagia is basically eating a lot. So that thing that you found to eat, in addition to putting in the mouth, you put in the mouth and you eat it a lot. Then we have hypersexuality, which is unusually heightened libido. People or animals which have this particular condition have been noted to have increased sexual desire even for across species, like in the image shown there, you can see a rabbit with a hen, for example. And the hen, the chicken may not have to be male, female can even be male. So basically some unusual heightened libido, that's hypersexuality. There's also visual agnosia, which is the inability to visually recognize familiar objects and familiar faces. So somebody you've seen before, you know them, but you can't recognize that visual agnosia. And lastly, we have hypermetamorphosis. Hypermetamorphosis is actually a irresistible impulse to react or respond to everything around you within sight. And this is a cause of distraction. So people with Kluver-Bucy syndrome are easily distracted by anything in the environment. Remember, it's caused by bilateral distraction of the amygdala. Maybe most of the symptoms were seen from experimental monkeys, but it's also been described in some people. Alzheimer's disease is another condition to talk about. It's a neurodegenerative disorder. We do not know the actual cause of Alzheimer's disease, but we know that Alzheimer's disease is a major cause of dementia. Dementia is decline in cognitive ability, and this usually impacts on the person's ability to perform day-to-day activity. There are many things involved in that. When you look at it pathologically, there's a strong association between Alzheimer's disease and accumulation and deposition of what we call cerebral beta amyloid. It's a particular protein that it's accumulating or depositing within regions of the brain, forming what we call plaques and neurofibrillary tangles. The eventual process is that now it will cause neuronal loss, so neurons die, and so there'll be volume loss in the brain. The regions of the brain which are affected in sequence are largely entorhinal cortex. This is part of the limbic system. Hippocampus, also part of the limbic system, and you can see that here. So this is where we expect the entorhinal cortex. Look at that severe atrophy. You hardly see any cortical tissue. Even this side, you hardly see any cortical tissue. Hippocampus, this side we see something, this side is really atrophic. We also have atrophy within the precuneal gyrus. So the precuneal gyrus is the gyrus in the parietal lobe near the occipital lobe. So you'd see it mainly on the medial aspect. And in this particular image, you can just see that there's some atrophy there. But we expect the precuneal gyrus to be much posterior to this. And lastly, The temporal lobe itself is usually affected as well. So the rest of the temporal lobe will also be atrophic. So, well, the atrophy will include many parts of the brain, but significant involvement of these regions. And we can see that we are starting some limbic structures. So in diagnosis, we look at a number of things. So mainly clinical diagnosis, but in terms of imaging, the imaging diagnosis that confirms or supports the diagnosis of Alzheimer's will be based on seeing atrophy on the mesotemporal lobe, as we can see here, or seeing atrophy involving both the temporal lobe as a whole and the parietal lobe as a whole. And in this case, you see the parietal lobe is significantly involved as well. Of course, there should be a clinical background to that. The other clinical condition is schizophrenia, which is a psychotic disorder characterized by distorted perception of reality and bizarre behavior. This is what some people call madness. Studies have shown that people with schizophrenia actually have reduced volume in the temporal lobes. And in particular, the limbic structures have reduced volume. And so that's why we are associating schizophrenia with limbic disorders. The other disorder I talk about is Korsakoff psychosis, which is caused by chronic thiamine deficiency. Thiamine is vitamin B1. And this usually causes atrophic changes in some parts of the brain, but in particular, mammillary bodies of the hypothalamus. Again, part of the limbic system, remember, part of the Papez circuit in particular. This is associated with memory loss and confabulation. Confabulation is the generation of false memory without intention of deceit. So people just say things which are really not true, but they describe them, and they're not really intending that to lie. This is a confabulation. It's usually memory loss. You've lost the actual memory and you're creating your own memory. Strongly associated alcoholism. The Korsakoff psychosis is the long-term effect of that, but the acute events would be things about acute memory loss and confusion. It may not go with the term Korsakoff psychosis. It may go with other terms. All right, thank you very much. I think we'll close there. We've quite taken a long journey through the limbic system, but we've covered the areas I want to cover. Thank you very much.